Yeah. So hello everyone, welcome back to our MLTB series of Meet and Learn with the Bioinformatician. This is the seventh installment of this series, and thank you everyone for joining this event. So first of all, I want to introduce our esteemed organization, ISCB-SC, RSG-India. As you guys know, ISCB stands for International Society of Computation Biology. So we are the International Society of Computation Biology. Our logo is your local window to global opportunities. So as we go further, so ISCB, ISCB-SC, and ISCB-SC RSG India. So as you know that ISCB is International Society of Computation Biology, under which Student Council came, and under this Student Council we have various regional society groups in 30 plus countries, and RSG India is among of them. So as you know that ISCB started back in USA in 90s, and then after that Student Council formed, and under the Student Council our RSG India stands. So for the RSG India, we have right now 4 RSG India nodes. There are various areas. They are situated in various states in our India, like in Hyderabad node, Mahindra University. In Karnataka, there is a Mahi Manipal Academy of Higher Education, and then there is Kerala node, NIT Calicut, and our new node which is started in Tamil Nadu, and this is in Sri Ram College. Then after that, these are our committee team members here, and the seventh installment. Before the seventh installment, we have 6 episodes with various esteemed bioinformatician from around the globe, like Professor Raja Majumdar, some PDB scientist, Professor Samrat Chatterjee. Then we have one of the main founders of Google Colab, Dr. Millet Medita. Then we have Professor GPS Raghava, and in our previous episodes talk was done by Dr. Harpreet Singh from ICMR. So these are some of the glimpses of those events. So I would like to welcome Professor Rajnish Kumar to come on stage and introduce our speaker, Dr. Rajnish Kumar.
So good afternoon, everyone. And first, I will thank you to come all the way here in this rainy season. So today we have a very distinguished speaker, Dr. Rajnish Kumar. So he is a professor at IIT-BHU, and by training he is a medicinal chemist. So nowadays everyone hear about AI and drug design. So basically he is the one who is using AI for designing drug. And let me say this, that he has several drugs in pipeline and in different stages of development. So I am not going to take more time for introducing him. Let's see what he has done till now and how drug is being designed using classical methods and new methods of AI. So he has also a SWAYAM course. If you want, you can join that also. So basically it's a audited course, so you can also take an exam and get a certificate of passing that. Okay, so welcome Dr. Rajnish to this forum.
I'm audible now online. So thank you so much, Professor Rajnish Kumar, for a kind introduction, and I'm really happy to be here, and also I would like to say thanks to the organizing team, Mr. Jatin, Mr. Govind, and other team members. It's really a privilege. Actually, it's my first time here at DU. I have heard DU a lot of times in my life, but some problems here. Okay, sorry for the disturbance. And thank you, madam, head of the department, for spending your time to be here. So today's talk is about just an introduction to what is drug discovery. Since this department is, I think, department of biophysics, and most of the researchers like PhD students or PI, they are must be working with drug discovery. So today I'll just give a glimpse of what is drug discovery and how we can use artificial intelligence in drug discovery. And then I will just give like a small example of how we have been using it in our lab, in our research. So it will be just take it as a like very basic, introduction to the topic actually. And I have been fortunate to work at Karolinska Institute, which is responsible for giving or awarding the Nobel Prize in Medicinal Physiology. When Alfred Nobel wrote his will back in around 1900, so he gave this, opportunity to Karolinska Institute to decide to whom they shall give the Nobel Prize. And this is the place called Nobel Forum, where, it's a kind of a building, small building, and they have auditoriums like this, where the Nobel Committee, which consists of 50 members of Nobel professors. So they all are professors at Karolinska Institute, and they're called as Nobel professors. So they sit all of the year, and then they decide, doing meetings and deciding to whom they shall give the Nobel Prize. So can anyone tell me who got the Nobel Prize in Physiology and Medicine in 2024? No? Okay. Yeah, so, and I have been fortunate to take my bus in front of this building almost every day, every working day. So I did my PhD from Punjab University, Chandigarh, and that is what I was telling to Manish sir, that I had a chance to visit his lab, his ex-lab, because at that time he left in tech Chandigarh. So I visited his lab, and then I was postdoc from 2014 to 2019, almost like 5.5 years, 6 years, and I was fortunate to work as a JSPS summer fellow at Nigion, Osaka University with Professor Keiji in the Laboratory of Computational Biology, Bioinformatics. And then since 2019, I've been working at IIT-BHU in the Department of Pharmaceutical Engineering. So IIT-BHU Department of Pharmaceutical Engineering also has a great history, that it is the first college or the institute to start degree level education not only in India but in Southeast Asia. So back in 1936, when the country was still under the British rule, so PC Ray, Praful Chandra Ray, who established the Bengal Chemicals. So he discussed with PC Ray, and then they started this pharmacy degree back in 1936. So coming to the topic, what is a drug, right? So if we say, can I ask like anyone of you what is a drug? What do you think about a drug? Like almost everyone of you is taking drug, right? I usually have this kind of interactive session, so yeah, just what is a drug? What do you think about a drug?
Drug is basically a chemical substance that we take, and it can help us, and it can also disrupt if it's not in the amounts that it's supposed to be.
Okay, so basically this is a very small single sentence definition, but this single definition has 4 hidden definitions in it. So let us break it down. So a substance used to prevent or cure a disease or ailment to alleviate its symptoms. Okay, so whenever we get fever, right? So paracetamol we are using, right? Crocin tablet, Noro 650. So what is that doing? That is just relieving the symptoms of the fever, right? So fever is a symptom of. It can be due to exertion, it can be due to infection, it can be due to injury, it can be due to anything. But paracetamol, what it is doing, it is just relieving the fever. So it is called a symptomatic treatment. It is just relieving the symptoms. Okay. On the other hand, if the fever is due to infection and we take antibiotic like ciprofloxacin, so what that drug is doing is that is curing the symptom. Curing the disease means that is killing the infectious organism, and that is curing the disease. That is called as disease modifying treatment. Okay, and then for example we take vitamin B12 tablets or iron tablets. Why do we take those? So that we do not get the disease. So it's a kind of a preventive, and that is called as prophylactic treatment. Okay, and then we have therapeutic, where once we get the disease, like someone has got hypertension, diabetes, cancer, right? So once we get the disease and then we take the drug, that is called as therapeutic. So now we have 4 definitions: symptomatic, disease curing, therapeutic, and prophylactic. I hope everyone just understood what is that, right? Now,
Please carry on. No problem.
it's not moving. It has to be on the.
Full screen.
Yeah, sure. So you just have to switch it. Now you don't see that. Are you happy with this view? No? Okay, then maybe I just do one more trick. Okay, okay. So you sir, you fix it. I fix it now. Okay, fine now. It's okay. Thanks. Really sorry, a lot of trouble I'm creating actually. Duplicate it and then I keep my cursor here. Yeah. Okay, sorry for all the trouble, right? So then the drug discovery has two parts. One is called as discovery, another one is called development. Okay, so in the discovery phase, whenever we are saying discovery, so you have you have to identify a target, and then you have to perform high throughput screening to identify those small molecules which can target that target, you can engage that target. And then you identify the inhibitors. Okay, you have to validate them because they might not be soluble, permeable, stable, metabolically stable, all those problems they will have. And then you get a lead compound which is a perfect compound which is soluble, stable, every good quality, followed by testing the preclinical toxicity studies, and then you get a drug candidate. So I usually give this example like you just lost a key to a log and you have to identify a key. So what you do is you just collect all the keys which are available with you, maybe 50,000 keys, and you try each and every one, okay, in the log, and some of the keys, one key out of those 50,000 that can open the log, but with some hurdles. It's not very smooth and easy, and then you just polish it, make those slots like bit bigger, smaller, and then you adjust, and then you can make it a good key actually to open that log. Right? So the challenge is that it is like the success rate is very low, the cost is very high, and it takes a lot of time for this discovery phase. Likewise, development phase is when you have once you get this drug candidate, that has to go IND enabling studies where you do detailed toxicity studies in at least 2 animal species. Mutagenicity studies, detailed pharmacology, acute toxicity, pharmacokinetics, all those studies you have to perform, and then you have to study the manufacturing processes as well. That since once it will be approved as a drug, it needs to be produced in tons of the quantity. It's not like in lab scale we are just producing milligrams, gram scale, but in clinical use you have to generate it in tons. So do we have a procedure, a GMP, good manufacturing practice, approved procedure where we can produce tons of that key API? So all those studies we have to perform, and once it clears all these hurdles, it move into the clinical trials, where the drug is now being tested not in animals but in humans. Okay, and it is only possible when the drug has been approved or drug has cleared all the animal experiments, that it has not shown any toxicity. It is very good solubility, very good bioavailability, permeability, all those pharmacokinetic, pharmacodynamic parameters. Again, the issue is that the success rate is very low, cost is very high, and it takes 4 to 6 years. So then what we can do with the help of AI? What we can do is we can use AI at each and every step of this drug discovery and development, and that is what I will show you today, like how do we do that. Okay, just to give you a real example, this is an example of drug called omeprazole. How many of you know what omeprazole is? Has anyone of you ever taken omeprazole?
It's an antacid.
Yes, almost everyone of us has taken omeprazole once in our life. Whenever you feel like acidity, hyperacidity, gas, so you go to chemist shop and then you ask that, tejab wali tablet capsule de do. Just to say, if you do it like this, it will rattle actually, because it has small granules in it, this hard gelatin capsule. Okay, so what is that drug doing is, so that is a proton pump inhibitor, decreasing the acid secretion in our stomach. So the company AstraZeneca, started their expedition to discover a drug called anti-secretory drug. So they identified this compound CMN131, which was very good at inhibiting the acid secretion. But when they tested it further, what they found was that this compound was showing liver toxicity. So they could not use it, because if compound is toxic, then you have to abandon it. You have no other choice rather than to change your strategy. Okay, then they came up with another compound that was a derivative of this compound, pyridine derivative. But then they found that this compound was already patented by some other company. Okay, so then also they cannot use that structure. They have to identify new compound. So then they came up with this compound, which was a metabolite of H124/26, where you can see that this S got the SO oxidized. Okay, and now this compound was again having a severe side effect where it was inhibiting the iodine uptake, right? And that is a serious toxicity. And further, they modified it, and then they came up with this compound called picobrazole in 1976, and that too was causing a lot of issues, having severe toxicity in the form of necrotizing vasculitis. So you can see, like, starting from one compound and coming up with new lead molecules, and all those molecules were either having toxicity or some other issues. So it was a big challenge. And finally, in 1979, they identified this compound, which was potent and non-toxic, and they named it omeprazole. And that became a blockbuster drug. Like we have blockbuster movies, so blockbuster drugs are drugs which has annual sale of at least $1 billion. You can just imagine the revenue this drug has generated. So they can change the life of the company, the country itself. Like this anti-obesity drug discovered by Novo Nordisk company. So it has a contribution of 3% of GDP in Danish economy. Just one drug molecule, sale of one drug molecule is impacting 3% of economy. You can just imagine like how big market this is. Okay, and that is why every big company, whether it is Microsoft or Google or Schrodinger, they are moving into drug discovery, and they are trying to develop new molecules for diseases. So so coming to the machine learning, right? So I just go quick. So what would be the next number? If I ask you 10, right? We can easily tell. Okay, but can you answer this question? The question is, which of the following molecule can cross blood-brain barrier? Can you answer this question? If you can answer, you say yes. Otherwise, you just say no. No? Why? Why? We were very good at answering this question, right? We could answer this question, but why can't we answer this question? Young student of chemical computational biology.
Our neural power is low compared to air.
No, I'm just asking. We are able to answer this question, right? Every one of us is able to answer this question, but why can't we answer this question?
There are so many benefits.
In simple words, if I say that we are not able to identify the pattern. Can we say that? Here we are able to identify the pattern that every time we are adding 2 into the number, previous number, and we are getting the last number, right? But here it is difficult for us to identify the pattern. If there is a chemist who is working for his or her whole life, like almost like 30 years in drug discovery, he or she can answer it with some good reliability. But otherwise, for us, we cannot, because we cannot understand the pattern, right? And with increase in the amount of data, like when we have thousands or billions of or millions of billions of those molecules, it is very difficult for us to identify the pattern. Where the AI can do it or machine learning can do it. Not only because they are smarter than us, they are not smarter than us. The only reason is that they are fast. They are fast, okay, and computer work while man is sleeping, because they can work 24 hours, right? Because then we have to take rest, okay. So they are faster, they can analyze, they have they have large memory storage, they have higher processing speed. That is why they can identify the pattern. It's not that they are smarter, actually. They are nowhere smarter. Only thing is that they are faster and they have more capability, storage capability, data processing capability. They can identify all those things. So that is where we can get the help. That is where AI can help us. Okay. Now let us see. Yeah. So this is a basic, summary of AI. So where everything is AI, and AI just is mimicking the natural intelligence. Right? What is intelligence? Do we consider ourselves intelligent? Yes or no? Yes. Almost every one of us consider ourselves intelligent, right? Why do we consider ourselves intelligent?
Because we have power of critical thinking.
Critical thinking. Okay.
Logic, reasoning.
Reasoning. Okay. Good. Anyone else?
Pattern identification.
Sorry.
Pattern identification.
Pattern identification. Decision making. Okay. So in simple words, for example, I have this water bottle, right? And if this water bottle is, it is containing hot boiling water. So what I will do is I will touch it and my hand will sense it that, okay, it is very hot. So I will decide not to drink it because it will burn my throat, right? So it's like you can collect the data, process it, and take a decision on the basis of that process. So this is basically, we can just define it as intelligence, right? And if we can make a machine or we can make an algorithm which can do the same thing, that is artificial intelligence, right? So everything is artificial intelligence, and a subset of it is machine learning, where we use a logistic regression, and a further subset is called as representational learning, where we use the autoencoders, and a further subset is called as deep learning, and it is deep buried within AI. That is why it is called as deep learning. So we have the deep learning where we use the multi-level perceptrons or artificial neural networks, where we do not have to explicitly code them, we do not have to give them direction. They can automatically analyze the data and take a decision on that, like self-driving cars. Okay, self-driving cars, or the robotic vacuum cleaner we have nowadays in our homes, right? So you just plug it into, it can automatically plug itself into the power plug as well when it needs charging, right? So basically, machine learning is we use the unknown data, we use the known data, and then we identify the trend in that data, and then we predict the unknown data. So this unknown data could be missing data, unseen data, data which is not existing, or it can be the future data. Like we have the weather forecasting app in our mobile, where we can see whether it will be going to rain for next 10 days or not, right? With a probability, like 60%, 70%, 30% chance of rain. What do you think? How does it calculate? Model, right? How does that model is built?
Data from past years.
Yeah, so Indian Meteorological Department, they have placed like sensors in several places in like Delhi, for example. The sensors are collecting data like humidity, relative humidity, wind speed, wind direction. Here the satellite images where we can see where are the clouds are moving in which direction, how much is the cloud density. So putting all these features, we call them features, putting all these features into a mathematical algorithm and identifying a pattern. And based on that pattern, we can predict whether the outcome actually, the future outcome. Okay. So coming back to the drug discovery, how we can use AI to accelerate the drug discovery. So basically, we can use it. It's not comprehensive actually. I just like given main things. So the first thing is that we can use it for target identification validation, right? Where we can do the protein ligand interaction prediction, or we can we can prioritize the target using the AI algorithms, or we can perform the high throughput screening, right? Where we can screen not millions but billions of compounds. During COVID time, several companies, for example, a group in US called as Holding at Home. So they screened billions of compounds against several COVID drug targets, and they identified several lead molecules. So that is where we can, screen billions of compounds and identify the hit compounds. And then we can use generative models, right, where to generate new structures even, and then we can use predictive toxicology for predicting the ADMET properties, and we can do the toxicity modeling. And not only that, we can also take help of AI in clinical trials, where we can design the clinical trials, where we can predict the outcome of clinical trials. Right. So let us go through them one by one. Jatin, please remind me how much time we have left actually, because I don't want to waste your time more than the limit. So coming to the target identification, so this is one recent study where the company called ISCB-SC, what they have done is they have, by taking into consideration the multi-omics data, proteomics, metabolomics, transcriptomics, epigenomics, all that data, combined with electronic health records, combined with clinical trial documents, combined with publication data, patent data. So they have screened all that data, and by using all that data, using their proprietary tool called as Pandaomics, they could identify a new drug target for a disease that is this idiopathic pulmonary fibrosis. Okay, so what they did was they took all this data, and then they did the novelty assessment. They identified some relevant targets. So they prioritized 20 targets based on all these type of data, patent, publications, clinical trial, grant data, and then deep learning based target ID engine they used. Okay, and then they identified 20 targets, and out of those 20 targets, they prioritized some of those targets by even wet lab validation, and then against those targets, they generated compound by using generative AI, using their proprietary tool Chemistry 42, and then these generated molecules were showing nanomolar affinity. And now one of those molecules, which is recently named as Rentosertib, so that is in phase 2 clinical trial. And this all they have done in very short period of time, just 18 months, from identifying a new drug target to phase 2 trial in a very record short time. In silico medicine, they have done it, and with the help of AI. This work, if it is done, otherwise it would take at least 5 to 8 years. Okay. Then how can we use it for the identification? So this is one of the study we also performed recently. One of my PhD student did that. So in this, what we did was there is this large, ultra large library of 1.3 billion compounds. That's a Jain database. So it is not possible to dope all of these compounds. Molecular doping will take, at least 6 months on a decent supercomputer as well, because you have to screen all those compounds. Each compound is taking for doping at least, even if it is taking like 10 milliseconds, it will be a lot of time. So what we did was we sampled, 1%, 0.1 to 1%, like 1 million compounds we randomly sampled from this library, and then we converted them into fingerprints, and then we doped these 1 million compounds and generated their doping score. Okay, and then we made a DL model using the fingerprints and to predict the doping score. So we made a decent model which can predict the molecular doping score of these molecules based on these 1 million compounds. And once we get this, so we use this model to screen the rest of the compounds, 1.3 billion compounds, and then we identify top hits. Okay, so this study is since we did not develop this tool is called as deep docking, developed by a Canadian professor. But since we used it, we modified it a little bit and we used it for our own. So this is the power of AI or machine learning for fit identification.
So in this case, your 2D fingerprints will remain the same always. Only your targets will change if we want to do it for anything.
Yeah, fingerprints will same, yes.
So fingerprint part will be always
same. Docking will be changed.
And that's why the DL model will change, and then you will get
exactly. So you can like, for example, once you can convert all these molecules into fingerprints, and then you can store it forever. You can use it for new targets. So for that, we just have to. But this is not just one cycle. You have to repeatedly do it. For example, we ran 3 iterations, 3 times, so that every time we are reducing the size of this library. For example, first time when we did, so this 1 billion was reduced to 100 million, and then 10 million, and then 1 million. So every time you are, giving it a cutoff that, okay, I want molecules only with docking score of minus 12.0. Okay, and then you just filter down that big large library and identify the compound.
What is the advantage of using this TL-based approach?
Yeah, so the advantage is because whenever we talk about virtual screening, so it is kind of an enrichment actually. So
like initially, I believe you took all the compound except you developed model.
No, we did not take all of the compounds actually. So you just take 1 million compounds out of this 1 billion
to develop the model.
Exactly, and then use that model to predict the docking score of all the molecules.
So once you predicted the docking score, you have predicted it for all the compounds.
Yeah. So once you, for example, once you got this model, and then you predict all these compounds, okay, and then, for example, you got 100 million compounds which are scoring above your cutoff limit.
So
right?
you just increase the cutoff.
No, and then you, because whenever you are randomly sampling those compounds, so whenever you are using it for this large library, so there are chances that it is not represented equally. So in the next term, when you do it again, so second time what you do is you again train your model actually on those remaining 100 million compounds, so as to improve the quality of your model.
Just like size blast.
Exactly. So you are in every iteration you are increasing the reliability of your model, right?
But then again, the overtraining phenomena comes in.
No, because, for example, now you did it, then you again sample 1 million molecules. So once you did it, you got 100 million. Okay, out of 100 million, you again select 1 million random sampling, and then dope them again, get their score, and then further again predict it. You reduce it to 10 million, again sample, you make a model, and then you do the screening.
Sir, you have taken a binary string 0, 1, 0, 1. So, sir, why didn't you take vibrational properties of a molecule?
Vibrational properties.
It can be a fingerprint.
Yeah, so for example, the fingerprints are, the one thing is that we can use molecular descriptors as well, because here we are just building a QSR model, right? Predictive model, right? And then you are converting your molecules into features. So those features could be molecular descriptors as well. But the problem with molecular descriptors is that those 2D descriptors are very less in number, and then you have to also generate the 3D descriptors, and for all those you need the structure in 2-dimensional format. And also for 3D descriptors, you need the structure in 3D format. Okay. On the other hand, these fingerprints you can directly generate from smiles. 1D, the 1D structure smiles you have, and from the smiles you can directly generate those fingerprints, and that is why it is fast and scalable. Scalable means for calculating molecular descriptors using Moodrad or Paddle library. If you want to calculate it for 1 billion molecules, it will take months, right? Because you are generating huge amount of data. On the other hand side, whenever you are generating the fingerprints, it's easy, fast, and scalable.
But that can be more accurate.
You're asking about confirmation diversity or vibrational
at the level of quantum biology.
Yeah, that's what I'm saying. Quantum is like for 1 million doing quantum mechanical calculation is a nightmare.
Calculation itself will take years.
Take years actually. So there is always, a trade-off between the speed and accuracy. So if it is, for example, quantum accuracy is very high, but the speed is very low. On the other hand, if you, reduce the level of this theory, for example, from QM you go to MM, molecular mechanics, so the speed will be very high. Further you go to coarse-grained simulation, the speed will be very, very high. So there is always a trade-off between speed and accuracy, and we have to settle somewhere actually. Based on what resources we have, what is our challenges.
So as you say, the moderate, you use moderate tools, so it generates 1800 properties of a single smile ID. So how can we minimize that properties? That which properties we should have taken for the?
In this case, we did not use moderate. I'm saying that if we use moderate, it will generate huge amount of data. It will take a lot of time, and that is why fingerprints are very fast and highly scalable. Okay. And then coming to the chemistry 42 tool from in silico medicine. So as I said, like they have identified inhibitors for DDR1 kinase as well, and you can see like the inhibitors they identified are in sub-nanomolar, sub-nanomolar potency. So you can generate molecules by using generative AI as well, and those molecules can be highly potent as well. Okay. Coming to generative AI, so if you ask ChatGPT any textual question, so it can generate a very nice text. Okay, it can generate very nice essays. But if you ask ChatGPT that can you generate molecules, can you generate some molecules which are BBB permeable or soluble or binding to some target, then it will start giving you yarn actually that BBB is like this, BBB is like that. But it will not be able to generate molecules. Why? Because it has not been trained on that kind of data, right? But there are tools which you can use to generate molecules. So one of the tool is so if we talk about de novo molecular design, it just automatically propose novel molecules based on the chemical structure, meeting the desired molecular profile. And then these can be trained on large data set to learn to produce output that is similar to the examples they have been trained on. Okay, so basically, if we talk about the molecular design approach, so there are two approaches basically. One is the direct approach. Okay, in direct approach, what we do is we have, for example, a set of molecules, and then we synthesize them. Like we synthesize one molecule in lab, and we test that molecule. Okay, and then we see that where does it fall on this property space. So this is property space, functional space we can say. Like you can say, for example, log P, solubility, and activity. Okay, so and your objective is that you wanted to have a molecule which is on the top of this hill, right? Which best molecule showing the best property. So one way is the direct approach is that you synthesize each and every molecule, you test each and every molecule, and then you see where it falls. And that's time-consuming classical method where you have to see each and every molecule, you have to test each and every molecule and determine its property. Okay. The other way is the inverse approach where you have all those the properties of these molecules in your hand, like we do in virtual screening, and then you just filter them down based on different criteria, and you try to identify your molecule of interest. Okay, that is the inverse approach. And then another approach is that is the generative AI approach, where you start with some point, start at some point. For example, we started with one molecule structure that is falling in this region, in this functional space, and now I try to optimize it to move it to this area, and finally aiming for a molecule which is falling in this area. Okay, so it's a kind of, there are multiple tools. For example, generative adversarial network, reinforcement learning, and then graph neural networks. So all those tools you can use to generate those molecules which are meeting your desired, specifications. And for that, this is a very good tool, Reinvent 4. million or 1 billion molecules, it can learn, and then it can just, learn how to generate a new molecule. Okay, so you have a generator model which can generate molecules on the basis of reinforcement learning, transfer learning, or just the atom by atom, generation. And then you have a scoring system which can be used for reinforcement learning. Now, what is reinforcement learning?
You make mistake and learn from those mistakes.
Sorry?
If you are doing something right, they will give you chocolate kind of, and if you are making a mistake, then
Yeah, so it's a reward and punishment, right? Reward and punishment model. For example, I am using a scoring subsystem where I use docking score as a scoring function. So every time, what it will do is, every time when this generator is generating a molecule, docking will be performed. Okay, and if the docking score is above the cutoff, like I need a molecule with docking score of minus 12.0. So if the docking score is below 12.0, minus 12.0, then it will keep those kind of molecules, keep on generating such molecules. So it will be a reward kind of thing. If it is generating a molecule with docking score greater than minus 12.0, like minus 10, minus 8, minus 6, so it will just drop those molecules because that will act as a punishment. Okay, so this is where the reinforcement learning is working, right? And then so it has all these generators, like it can generate molecules atom by atom, and then it can generate the molecules. For example, they can just generate the scaffolds actually. Like you have the molecule and you want to just replace the core scaffold, so it can just replace the core scaffold. It can just replace the functional groups, R groups. So all those sort of variations you can do. And it can generate the links as well. Like you have two molecules, you wanted to link these two molecules by a linker, so it can generate those linkers. There you can use the link event, and it has this mole-to-mole transport based on the molecular optimization. And then different scoring you can use, like USAR. For example, you can make a prediction model, like IC50 prediction model. You can put that IC50 prediction model, and you can ask that, okay, I want only molecules which are having this IC50 value less than 10 nanomolar. So every time this will generate a molecule, it will send it to the QSR model. It will predict the IC50. If it is less than 10, it will retain those molecules. If not, it will just drop those molecules, generate molecules which are not similar to that, molecules which are dissimilar to that. Okay, so this is how reinforcement learning is helping this reinvent 4 to generate molecules. And then you can have pharmacophore based scoring system. You can have ChemProbe. So ChemProbe is a kind of a library where you can predict multiple properties at once. Okay. Yeah. Coming to the toxicity prediction, then you can. There are multiple tools. Toxigate is one which is developed by Professor Kenji Mizuguchi, and then this is another tool which is TG Gates, Open TG Gates, which can be used. And then you have the SafeAt AI, which can be used to predict the basically three. Basically, you can predict three toxicity properties: DPV. D is drug-induced liver injury. Okay, deep CAR, carcinogenicity, and deep MS. MS is again indicating the mutagenicity potential. So in deep DD, they have just taken the structure of those molecules and used different ML algorithms, and then they could make a model which can predict whether a molecule is DD positive or negative. So it's basic predictive modeling, nothing more than that. So this is the workflow. I've skipped that, and then similarly they have made this deep learning framework for predicting the carcinogenicity. They have started with 863 compounds, classified them into active and inactive, means toxic and non-toxic. And then based on those, machine learning models, they could classify them into, whether a molecule is, carcinogenic or not carcinogenic. Likewise, there is this app called Skin Doctor, so which can predict whether the molecules are, skin sensitizer or not. So it's a classification model: skin sensitizer, non-sensitizer, and no prediction possible. These are very simple models which anyone can make, And then there is this deep hit, which can predict the Hutch-induced cardiotoxicity, where it uses three approaches: descriptor-based, fingerprint-based, graph-based. Like you asked, right? So why can't we use quantum mechanical? So here you can see, like, they have tried three different approaches. Where in the first approach they have used the molecular descriptors. In second approach they have used the fingerprints, right? the fingerprints which are having 1024 bit long fingerprints. And then in third approach, they have used the graph neural nets. Like you can use even graph theory to convert your molecules into graphs, which contains the nodes and as if, and then you can make predictive models on those as well. Got it? And then you have chances to use predictive modeling, predictive advect properties. And I've been fortunate to work with Professor Kenji Mizuguchi back in 2019 as JSP Summer Fellow, and they have been developing such a nice tool called DRUMAP, a drug metabolism and pharmacokinetic prediction tool. So it's a very nice tool. So they have keep on adding new and new properties every month. So in this case, for example, they have made a prediction model to predict the fraction of unbound drug in the brain. Right, and in this case, what they did was they used the Campbell database. They generated the descriptors using CDK, Moodrat, and Paddle, and they used different ML algorithms like random forest, gradient boost, SVM, radial functional kernel, and PLS, and they have validated their model on R-square and RMSE. So this is their the landing page. Oh, sorry.
There is a word predict fraction.
Predict
Fraction of unbound drug.
Yeah, fraction of unbound drug in brain. Fraction of unbound drug in brain. So see, what happens is whenever we are taking a drug, right? Whenever you are taking paracetamol, okay? So it's not like you are just dissolving it in a glass of water. Whenever you take the drug, it goes into your stomach. Okay, and from stomach it is absorbed in the blood, right? And that blood goes to liver, right? And that is called as first pass metabolism. It has to pass through the liver because that is first defense mechanism. Whenever we eat something, even the food is also the things which are absorbed from food also goes to liver. The blood from GIT goes through the liver. It is filtered there, and then from that blood it is circulated in the whole body. Okay. Now, there is a very important term that is called as fraction unbound. Why fraction unbound? Because that is the only molecules, drug molecules, which are available to bind to your target. For example, whenever your paracetamol is in your blood, okay, most of the paracetamol is still bound to protein called albumin. Okay, and it acts as a kind of reservoir actually. So it will be bound to albumin, and only free amount of drug that will be available to bind to your receptor or your drug target. So this amount of drug which is bound to protein, that is not active in nature. So if a drug is more bound to the protein, or it is more unbound, the fraction which is more unbound, right? Unbound fraction, if it is more, it means that you need less dose of that drug. Have you ever wondered why you need 650 milligram of paracetamol as compared to only half a milligram of this, what is called as, or 5 milligram of cetirizine? Have you ever wondered? I'm asking, have you ever wondered? Give it alprazolam, we just need 0.5 milligram, where we need 650 milligram of paracetamol. Why? Because of their properties, their pharmacokinetic and pharmacodynamic properties. So whatever a drug is doing to our body is called as pharmacodynamics, and whatever our body is doing to the drug, it is called as pharmacokinetic. Because our body consider a drug means anything which does not belong to our body will consider it as a foreign object, as a threat, right? So as soon as you take the drug, our body will start, getting rid of it, right? So either metabolizing it, excreting it through urine, excreting it through feces, right? Or just breaking it down into simpler molecules so that it just, goes away. It's very complex actually. So fraction unbound is, if the fraction unbound is more, then the effect will be more, and you need less quantity of drug. Clear? So this is an important pharmacodynamic parameter. Yes. Yes, please.
Could I please remind you that the time is 4:40?
How much time I have?
5 minutes.
We have a workshop.
Just 5 minutes. Okay.
20 minutes. Can I please request you to? We have questions.
Yeah, we will take question later. Actually, let me finish it because there are always multiple correct. So I'll be here for another 2 hours. No, don't worry. Okay.
Please remember and write down your questions. Okay. Please ask your questions to one speaker. Thank you.
Okay, so I'll go quickly because there are some just very few slides actually. So this is the TrueMap version. So this is now I think they have version 2.0. So I suggest that you just search TrueMap in Google and then you can get it. And then coming back to the clinical, the last thing which is the clinical trials. So how we can use AI in clinical trials? Okay. So just imagine in clinical trial what we are doing is we are just evaluating the drug molecule in humans. For example, we all are registered in a clinical trial. We all are participating in a clinical trial. So some of us will be responders. That is called as responders are those who are showing effect. Like if I'm measuring the blood pressure reducing power of a drug, so those who are showing the effect means their blood pressure will go down to a certain limit, and those who are not responding means they did not show any effect, or they did not show the effect up to that the cutoff. Okay, so basically it is responders and non-responders, right? What if I can by some way by computational modeling or by AI I can tell that those who are sitting on the left hand side will respond to the treatment, and those who are sitting on the right hand side they will not respond to the treatment. Is it a good or bad? Good or bad? Good, because if a drug is failing in phase 3 or phase 2 clinical trial, the company is losing billions of revenue, right? Because they have spent like 10 years, 8 years in developing that drug, and now this drug failed. Okay, so they have wasted all the money. So if I can, with confidence, I can tell that okay, these will be responding, these will be not responding, then I can, I can improve the chances of success of the clinical trial. And that is what AI can help us with. Not only because it is smart, because it can analyze the data, it can identify the pattern. So if I can take all the relevant details, like all the diagnostic data, all your proteomics, genomics, metabolomics, transcriptomics, epigenomics, all that omics data, all the patient history, and then I can fit that into a model, and that can tell, okay, these will be likely to be responding to the treatment, and I can keep those into that arm of clinical trial. It will increase the chances of success, right? So that is, and there have been several startup companies who are doing this actually. So this is a tool called PyTrial, which can predict the patient outcome. It can help you with the trial selection. It can help with the trial outcome prediction. It can help with the patient trial matching. It can help with the trial similarity search and synthetic data generation. I'll skip this. Coming to the limitation, everything which seems good has some limitations, right? So what are those limitations? Recently, we saw, not recently, 2023 news, I think, 2023. So that one of the drugs which was discovered using AI, it failed in 2 phase 3 trials. And you can just imagine like how much money they would have lost actually. So that is why drug discovery and development is very, very complex. It's not as simple as developing a classification model, cats versus dogs, okay, or identifying a plant just by taking a picture of it. It's not as simple as that, because there are several challenges. There are hundreds of challenges, and it can go wrong at any time, right? And then if you look at the Gartner cycle, the hype cycle, so we are right now at the peak of inflated expectation. Soon we will see the trough of disillusionment, and then we will hopefully see the slope of enlightenment and some productivity. Maybe in like down the lane in 1 or 2 decades, we will see some real impact. However, we are seeing it. We are seeing it now as well, but in maybe 1 or 2 decades, we will see the real impact of this technology. Coming to the real limitations, the limitations are the data quality and the availability. Most of the data which is available is with those big companies, right? Big pharma companies, and they are not ready to share it with anyone else, right? They are sitting on the data. So if there is no data, means there is no model, right? And in computer science, garbage in, garbage out. If the data is not good, your outcome will definitely not be better. So heterogeneity is another problem. The data which people are using is from the public data sets. The assays which are done in like different labs, they might have been done at different conditions actually. So we cannot just rely, we cannot just put them, club them all together. Complexity of biological system. Biological system is very complex. Diseases are multifactorial in nature, like the Alzheimer's disease, which has tauopathy, amyloidopathy, inflammation, and all those sort of things. So where we cannot simply define them in one single equation, right? Interpretability problems, the black box nature of the AI models. That is why we now see those XAI development of XAI, explainable AI, where people are trying to develop models which can have some explainability in them. I read it somewhere that the purpose of development of AI was to understand how the human brain works. Okay, but now the irony is that we don't know how AI is working. So that's a kind of big problem. Transferability of models. If I just say that I have made a model on 100 molecules to predict solubility of those molecules, can I apply this model on 1 million molecules? Yes or no? Can I do that? Yes? No. Because those 100 molecules, it's like, for example, just taking the height of all of us, or maybe like girls, height of these girls, and then saying that this is the national average height of girls. Can you do that? Because our, the starting point was limited, very limited. We have just trained our model on very limited amount of data. It is not generalized model. We cannot apply it to any other, So every model has its own applicability domain that it can be applied. You might have seen those terms and conditions on every advertisement, right? Terms and conditions. Like you get 20% cash back, limited to only 500 rupees. Amazon will take up to 25% cash back, limited to 500 rupees only. No matter you spend like 2 lakh, you will only get 500. And then computational resources for deep learning, especially you need like really aggressive GPU machines. Ethical and regulatory concern, you all have seen even the big leaders in AI, they have said that AI is, lot of issues. Recently, several countries, they have, banned some of those things. Collaboration and integration, lack of collaboration, industries are not ready to collaborate with academia, right? And then post and investment, again, yeah. And then I'll just give you some example how we have been using it, just maybe 2 minutes. So AD, we are working in the field of Alzheimer's disease, and Alzheimer's disease is the dementia where the brain starts to lose its connections because of the deposit of amyloid beta plaques and activation of microglia and deposit of neurofibrillary tangles. So the idea is what we are working is on early diagnosis and early treatment of the disease because that is the key. So in that line, we have worked on identification of BBB permeable polyacetyl transferase ligands, which can be developed as positron emission tomography tracers to diagnose the disease in early stage. Okay, because the current diagnostic agents, they are not specific to the disease and they show a lot of off-target binding. Okay, so CAT is an enzyme which synthesizes acetylcholine and it has this tunnel kind of shape where the histidine 324 amino residue is responsible for acid-base catalysis. Okay, there are several derivatives reported, but none of them are BBB permeable because of their structure, mostly because of this quaternary nitrogen containing compounds. So what we did was we did kind of a virtual screening of libraries and we identified several compounds like you can see here. This is one of the compounds which is having nanomolar affinity. And then we have screened another library around 1.4 million compounds. And then we have got several nice compounds. So after purchasing them and testing them, we could see that they are really good. And then some of them are really, giving nice activity and kind of a mixed kind of inhibition. Okay. And then the beauty of this is that they are structurally they are novel. So they have not been reported And novelty is very important, especially when you are after the drug development, because you have to get the patent, you have to secure your rights. Otherwise, you cannot use the compounds which have already been reported. Okay, and then we are making them more better actually by derivatizing them. So, for example, in this case, we synthesized a lot of derivatives of this compound, and then we came up with better, compounds with better solubility, better permeability. And then, as I said, like I was discussing with Madam, that we have been using single particle cryo as well, cryo as well. So you can see here, like these are the molecules. Some of the molecules which are showing the these are, you can see like dimer forming the dimers. We collected this data, and then we try to resolve the structure. So this is 3D reconstruction of the cholinesterase transferase, and the idea is that we have a ligand. If we can see how it is binding, we can modify it on the basis of how does it bind, and that is called a structure-based drug design. That is we are doing. And then, as I discussed earlier, that we have used this approach, deep doping approach, to screen 1.3 billion molecules library, and then we could identify some of those compounds, like these compounds, which are showing better activity, and now we are purchasing them and we are evaluating them. Okay. Yeah. So, in conclusion, we can say that AI is highly promising in accelerating drug discovery development, not only in the hit identification, but target identification, optimization, admit prediction, and even in clinical trials. And it can increase productivity and reduce cost by reducing the time taken, okay, and by increasing the speed. However, there are a lot of challenges, like with the data quality, and challenges with the interpretability, and also with the availability of resources, the hardware resources as well. And this is the course which I have recently floated on NPTEL, and the response is very good. Already 10,610 students they have registered. Not only students but faculties from different colleges, and almost 200 people they have registered for exam as well. So unfortunately, this is not open now for registration, but maybe those who are interested, they can register it in next semester as well, because I hope that it will run in the next semester as well. Okay, with that, I'm done, and we can start with this small exercise on Colab, which you can do with your laptop or with your mobile even. With your mobile phone. Okay.
So everyone, those are the DU students, they can use my DU Wi-Fi, and if they are not a DU student, they can use only DU named Wi-Fi. The password is in lowercase, admin@123. Admin@123. ADMIN@123.
So DU students can use the my DU login. It will work.
They've answered all the questions that we have asked.
Okay. Yeah, meanwhile you can have questions actually. Meanwhile you can have questions. What happens with the if we are using these tools and generate a new molecule, so there is no problem, because IPR we get for the molecule actually.
So like so I had a very long discussion with Auditorium, and they were trying they are purchasing academic license. So when I asked that, let's suppose I design it going further, so then they said that if this license, you can't go for commercial license. So that is that
because this is live session actually, yeah. But there is nothing to worry about that.
I had a question.
Yes, please. Anyone? Yes, please.
So my question is about the data set you are using. So like how you are verifying this particular part of the data set is actually accurate or you and my second question, so that you can answer together. My second question is, like, are you also using chemistry 42 in the project that you are currently working?
Yeah, thank you. Sit down. So your first question is data, right? So as I said, like data is the king in computational modeling, and we have to make sure that we have to spend 80% of our total time on just data curation. So we make sure that okay, we manually curate the data. We do not rely on the data which is curated by someone else, for example, and then we curate the data manually, or you use the reliable sources. Like you can use the Campbell, you can use the Bioinformatician, you can use the Auditorium or Jain. So they have they have their data policy where they curate the data and then they review it and then they only release the data.
So is this open source or like?
Yes, all these are open source. And coming to your second question, chemistry 42. So personally, I don't go for the license thing because of course working in academia has its own limitation. We don't have unlimited resources, so we cannot spend lakhs of rupees on license. So right now we are working with open source tools, and open source tools are also as good as the paid tools. The only difference is you go to Cafe Coffee Day or you go to this India Coffee House and then you ask them and they will serve you on the table idli sambar. The other thing is you go to buy rice, you soak it overnight, you grind it and you make your own idli. That is the only difference. If you are good at making idli, you can make better idli than you get at the India Coffee House. But if you are not good at making idli, you can make a worse idli. So you need to be efficient. You need to be good at what you are doing, and that can be done with open source tools. And all of those mostly nowadays, all these companies like yesterday companies, they are releasing their codes on GitHub, so you can use them freely. Like on you can get all those models, transformer models, LLM models, everything you can get there. Only you have to learn how to use them, how to make use of them. Any other please?
Sir, you mentioned about the unbound drug component there. Is that evaluated through the admit analysis only or through some other procedure?
No. Whenever you are making a prediction model, the data which you are using to make a model need to be experimental. You cannot make a model on predicted data. So as I said, like weather forecasting, you cannot just rely on your weather app. For example, for next 10 days it is showing that it will rain. You cannot use that data to make a model. The data which you will use to make a model will be experimental data, which all those sensors have covered, isn't it? Right? So whenever you are making a model, you have to collect the experimental data, make sure that the data is correct, and then only you can use that data to make a predictive model. Okay, so everyone is has opened that. So whenever you are okay, so I stopped sharing that and I share this one instead. Okay, so are the online audience following me? So is every one of you at this stage now? Can you open that? Okay. So here I have made a kind of quick predictive model where you can predict the bioactivity of any target, for example. Okay. So I will quickly go through it, and then because this is really available, you can open it anytime. It will remain with you, and then you can play with it actually. So the only thing you have to do is you have to make a local copy into your drive. So you just go to File and save a copy in Drive. Go to File, save a copy in Drive, because you probably will not be able to run it because this is posted in my Google Drive. So you just have to make a local copy. Go to File, save a copy in Drive.
If any one of you is on BU email, download from that because email, Google Drive, that was supposed to that.
Yeah, your own, your personal. Just log in with your personal
personal
Gmail.
Gmail account.
Go with your personal Gmail account. or you can just scroll it through and you can do it later. It's not necessary that you have to do it now actually. So I'll just quickly explain it.
Recording will be available.
Recording will be available on YouTube, so you can also just watch how each run is, and then you can run it.
After 30 minutes.
Okay, have you done it? All of you? At least those who are running it. So you just make a copy, save a copy in Drive, and when you save a copy, so what it will show is, it will show something like this. Yeah, so you can see here, copy of 12.3 bioactivity prediction. So you will see something like this actually, right? So now to save time, I will just you can do is you can just click on run time and run all. Okay, so that's the the beauty of Colab, that you can make a pipeline and you can run all the codes at once. So just run all, or from runtime run all. Okay, and then I can explain quickly what we are doing. Okay, so the first thing is we are installing RDKit. Okay, how many of you have opened this page? Yeah, okay, good. How many of you have copied it? Made a local copy. Okay, good. So if you have neighbors who are unable to do it, just help them. Just do the teamwork. Help your neighbors to achieve it. Sorry. Are the online audience able to do it?
You have to just click on that square bracket, install RDKit, square bracket there.
Okay, are you able to run the first cell, for example, install RDKit? Okay, so we do it like this. So I just explain it, and then you go home and then you do it, right? Because to be honest, running this whole tutorial, it needs at least like 2 hours actually to do all the steps. But just try to understand it, what it is doing. I'll just explain, and then because this will be available with you, so you just go home and then you do it. Okay, is that okay? Yeah. So because we just had 20 minutes actually. So just look at this section, look at the screen, right? So what we are doing here is we are trying to build an SVM based classification model, support vector machine based classification model to predict the bioactivity. Okay, and since the bioactivity data we will be taking from Campbell, so and we are using RDKit for doing all the modeling, most of the modeling. So we install the RDKit first. Okay, once you run the cell, so you'll see that RDKit will be installed, right? And after installing RDKit, you install all the required packages, like you install the Campbell web resource client, pandas, and then you import all those modules where we have installed, and then here you can see that I have taken the selected targets. For example, okay, so this is H is not correct actually. So the target ID is not H here, so it should be for MAPT. So if I open, for example, let me share the whole screen instead. Yeah. So if I open the so this is your Campbell database, right? And then in Campbell database, have you ever seen the Campbell database website? Some of you? Okay, good. So the beauty of Campbell database is it has the structures, okay, along with their biological activity. And how do they curate that is they curate all the data from published literature, patents, and all. So they have automatic crawlers which collect the data and then they curate it. Okay. So from this Campbell database, we can get for example, the target. Like if you search MAPT, microtubule associated protein tau. So when I say MPT, type MAPT here, so you will see the MAPT. So if you go to the target, okay. So you have the 2-1-0 target, and then you can, for example, you can see like these are from rat and then humans. So we are interested in the human. So we can select from here like the Homo sapiens. Okay. And then from Homo sapiens.
If you are not understanding anything, you can ask sir, and the recordings will also be available after 1 week of the session.
Research MLTB should. But it's okay. I think this is 312. So the idea is that we get the compounds from this microtubule associated protein Homo sapiens. Okay, here is it. Here it is. Once we get this, so now we will identify. We will see what are those small molecules activity chart. Yeah. So you can see here there are 94,769 compounds. So do you see that? 95,548 compounds. So out of them, they have IC50 data for 267 compound, inhibition data for 257, activity, KI, DC50, KD, potency, EC50. So all these are the compounds which have been deposited in this database in the name of MAPT, microtubule associated protein tau. So what we will do is we will just take the ID of this target. The target ID is, yeah, so this is this 1293224. Okay. So we just copy this and then type it here. Right. So when we type this selected target, so this is our target, and this you can replace with your other targets as well. So if you wanted to, for example, make a model on any other target, so you can replace it with any other target ID as well. Got it? So once you do that, so what we are doing is we are selecting the target. We have identified, okay, this is our target, and then we pass the IC50 data. Okay. Once we pass the IC50 data, so this code, what it is, it will make a data frame where it will extract all the data from the Campbell database for this ID, for this protein, for this drug target. Okay, and then in the next step we clean the data. So what we are doing is we are just removing all those columns where we have the NaN values, right? And also we are only keeping the standard values, and then we are fetching the SMILES as well, SMILES and Campbell ID of those molecules, and we are getting the molecular structure in the form of canonical SMILES. Okay, and after that what we are doing is we are now, once we have fetched the SMILES as well as the activity data, so now the next step is that we have to make sure, we have to classify that data into active and inactive. Okay, for example, if we wanted to make a model to classify cats and dogs, so we have to label the pictures, that these are pictures of cats, pictures of dogs, and that is called as supervised learning, right? Supervised learning, where we have labeled the data. Likewise, here we are labeling our data that if the value of IC50 is less than 1000, I am saying that these molecules are active. Okay, and if the value is more than 10,000, I'm saying that these molecules are inactive. Okay, and there is a margin between them, and that we have to give actually, because if we keep a very how to say, very strict boundary, so then our model will not be good actually. So we have to give some margin to it, that okay, less than 1000 is active, more than 10,000 inactive, and those are which are falling in between, we just neglect them. Okay, they are neither active nor inactive. Okay, so once we have classified them, so this is called as labeling. Once we have labeled them that these are active, these are inactive, the next step is we drop the NaN values and we just keep the columns which are relevant actually, and then we can just count how many actives and how many inactives we have. Okay, and then we can just see by this command called df.head, we can just see top 10 entries into our a data frame. So now you can see here, for example, I have these molecules. So where I have the Campbell ID, I have the smiles, the 1D structure of those compounds. I have the IC50 value. The value is, for example, 256. So I have labeled it as, active, because it is less than 1000 nanomolar, the IC50 value. Likewise, if I just issue the command df, which will give me a summary of the first 5 compounds and last 5 compounds. So you can see here, the molecules which are having IC50 value of 15,000, they are labeled as inactive. Got it? So we downloaded the data, we classified the data into active and inactive. Okay, and then after that, what we are doing is, now the next thing is to convert these smiles into RDKit mole objects. Okay, and you can just have a look by issuing this command mole list 1. So you can just have a look at this structure of the first compound. Got it? And then you can see like how many molecules are there. We have 182 molecules, and then we convert these molecule list to Panda data frame. Okay, into a data frame called df_ml. So now we have converted these smiles into RDKit mole object. The purpose of converting them into RDKit mole objects is because we need to calculate the fingerprints now, right? So whenever we are using SVM, we will calculate the fingerprints of those molecules. So because RDKit cannot read the smiles directly, it cannot calculate the fingerprints on smiles directly. So we have to convert it into RDKit mole object, and we can just visualize them as well. Okay. So in next step, what we are doing is we are using the RDKit fingerprint generator, which is a function in the RDKit to generate the fingerprints. So here you have several possibilities. You can use like multiple. For example, this fingerprint here we are using is called ECFP, eccentric connective fingerprint. It is just based on the connectivity of how those atoms are connected to each other. Okay, 1 layer, 2 layer, 3 layer. So we are just converting a molecule structure into numbers, a string of numbers, binary numbers 0, 1, 0, 1, based on how those atoms are connected to each other. Okay, so and this is 1024 bit long. So every molecule we convert it into a string of 1024 bits, where there will be 0, 1, 0, 1, 0, 1, 0, 1 based on their structure. So it's like we have our fingerprints. We have converted a molecule into numbers which can be read by the computer. Okay. So once we convert them into fingerprints, then the next thing is we convert this the fingerprints into a data frame. Okay. And then we display the data frame. So if you see, so this is now you have the eccentric connectivity fingerprint which we have generated in the form of 0, 1, 0, 1. Okay. And then you can just label them as the X matrix. So in predictive modeling, we have two things. One is called as dependent variable, another is independent variable. Okay. So dependent variable is usually our bioactivity or our target value, which we are trying to predict using the model, or which we are making the model on, right? And the non-dependent variables are the features actually. So here our X matrix is the features, those fingerprints, those independent variables which we have calculated from this line. Okay. So that is our X matrix. Now we have made an X matrix, and then we have made a Y matrix, which is our activity data. So which we recently assigned to all those molecules 0, 1, 0, 1 based on the IC50 cutoff. Got it? So that is our activity data. So this is our 0, 1, 0, 1. That is our, whether the molecule is active or inactive, the label on those molecules. The next step is splitting the data into test and training set, right? So now usually what we do is we split it into 80-20, like 80% of data we keep in the training set, 20% we keep in the test set. And in best cases, we can have an external test set as well, like we can have 60-20-20, 60 training, 20 test, and 24 validation set, which is not being used in building the model. That is the best case scenario. And once we have split this, So you can see like here we are using the X train, Y train and X test, Y test, where we are splitting it into 0.20 means 20% we are keeping in the test set and 80% we are keeping in the training set. And the next step is making the model. So here we are using SL classifier. Okay, by using SL classifier, we can just quickly make a model. And here, once we have run the model, made the model, we can use this model to predict the values of the test set. Okay, and the training set as well. And if you look at the accuracy of this model, so you can see like the accuracy is 0.91, means out of 100 predictions, it is good at predicting 91 of them as correct. Okay, and this is confusion matrix. So in confusion matrix, I think I have plotted it here as well. Yeah, so I have plotted the confusion matrix here as well. So here in this in the top right corner, so you can see this is your true label and this is your predicted label, right? So predicted label is 1, for example, here, and then true label is 0, means it is inactive. In this case, this molecule is the class 0, means it is inactive, and then this one is the true label, means the molecule is originally inactive, right? And then but it is predicted as active, so it is called as false positive. The top right corner of confusion matrix is indicating false positive, means those molecules are inactive but the model is predicting them as active. On the other hand side, the bottom left corner, it is showing as opposite to false positive. What will be opposite of false positive? True negative, because this is active, but the model is saying that it is inactive. Okay, right? So in this case, and then if you have a kind of a biased model, so what you will see is you will see here load of number, here load of number. So now you can see almost like similar. So you have a kind of a balanced model actually. If your model is biased towards positive or biased towards negative, you will see an imbalance in that. So the idea is that you should have least molecules here, and then most of them should be here actually. And then you can plot the ROC curve as well. And in this case, if you look at this ROC curve, so you can clearly see that if the ROC curve is close to the diagonal, it is worst, means it is just 50-50, showing 50-50% chances. But if it is close to this top left corner, then it is the ROC curve is good actually. It means that it is predicting those molecules very well. Those positives are actives are predicted as actives and inactives are predicted as inactives in most of the cases. Okay, so that was like model building. Now the real use of model building is using this model to predict new molecules. Okay, the activity of new molecules. Since this was a small data set, why? Because originally in Campbell they had 96,000 compounds. Okay, but out of them they only had IC50 value for around 250 molecules. Okay, so our model is not built on 96,000 compounds. It is only built on 250 molecules. Okay, so that is very important. So now if we talk about the applicability domain of this model, so this model is very limited to all those structures relevant to those 250 molecules. Okay, so now we can save the model. We can save the model in the form of the PKL, svm_model.pkl, and then now we can use this model to predict the activity of new molecules. So new molecules we will get from one of our GitHub repo. So there is this CSV file which contains just the Campbell ID, the smiles, okay, and the bioactivity which is not related to tau actually. So this is not tau activity, this is ACHE activity. So we don't have to bother about it. So we just download this, import this file, clone this file from the GitHub repo, and then we will use the structure, and then we will predict their activity towards tau by using the model which we have just saved, right? Now, who will tell me what are the steps how we can predict the property of these models or these molecules, or what we have to do? We have made a model, we have saved that model. Now what we will do? If I want to predict, how will superimpose? Yeah, validate we have already done, for example, with the test set. That's the final model we can say. Now we are just predicting the new molecule. So what I have to do is I have to just calculate the fingerprints, right? Because in that model, what did I use? Fingerprints. So I just put the fingerprint. That was my X matrix, right? Dependent, independent variables to predict the dependent variable Y. So if I can calculate the fingerprints of these new molecules and use this as X matrix to predict its Y, I can do that by using that model, right? It's like just calculating those features and putting those features into that model to predict the activity. So then I will use again the RF generator, RF fingerprint generator, convert these into fingerprint, okay, save it as a, the X matrix. Here you can see I have saved them as X matrix, X underscore ACHE, and then I import that model, okay, and then I just save the outcome of this model, which is predicted X underscore ACHE, and then here you can see the outcome is like this. You have the model, you have the Campbell ID, smiles, the bioactivity which was already there in this CSV, and the predicted tau activity. So here you can see like some of those molecules are predicted as 1, which is active. Some of them are predicted as 0, which are inactive, right? So this is pretty simple actually, and you can convert it into a web app as well. You can just code it in HTML using like React.js or any other framework, and then you can just make it a web app where anyone can upload their structure, okay, or enter their smiles, and then this model can predict whether that molecule will be tau active or tau inactive. Got it? Yeah, and then you can just save the activity prediction into CSV file, and then you can just open it in any other tool as well. And then in the end, I just show here another library which is very interesting called Lazy Predict. So instead of only building one model at a time, Lazy Predict can help you to quickly build around 30 to 40 different models in a very short time actually. Okay, quickly. So like you just install the Lazy Predict. Here you just keep install Lazy Predict, and then you use the Lazy Predict and use the same test string split, and then you can predict, fit and evaluate the models. And here you can see that it has made several models like using perceptrons, nearest centroid, random forest classifier, logistic regression, SDG classifier, rich classification, extra tree, and you can see the accuracy, ROC, AUC, F score. So all these parameters you can see, and the model, for example, now you can start with fine tuning or hyperparameter optimization for these top performing models. So it is a very quick way to just see called as prototyping, you can say. You can quickly see which of those algorithms are working, giving you a good accuracy, and then you can pick that specifically and then train a model, optimize the hyperparameters to get a nice model. Okay, so that was all actually. So is it clear, this quick hands-on? To be honest, as I said, like it needs at least 2 hours to do it in detail. But since it will be available with you, and you can just, the Campbell ID, you can put any Campbell ID of your interest, just play with it, run it. The code is very simple and very friendly actually. Okay, any questions from the hands-on? Are you on time, Jatin? Are you on time? Any questions from the online audience?
question.
Sir, I want to ask you.
Yes, please.
So, we generate negative data also. We keep that negative data somewhere, or we just put it into trash? Because sometimes the negative data contains a lot of information.
Which negative data?
Like false data.
No, that zero is also being used, right? So, of course, we cannot just train on the positive data actually. For example, we had 250 molecules, 100 we classified them as active, 100 we classified them inactive. We are using both those 100 sets to train our models. It's not that we are just using those 100. It's like if I'm training a model on cat versus dog. So if I'm using just images of cat, then everything which is not cat will be considered as dog, right? And that's not good. So we are using the zeros as well. We are using the negatively labeled data as well. Any other question? Okay, so someone is asking like
they are collating the question.
Okay.
Hello.
Yes, you are audible. Actually, you can please ask.
Yes, sir. Can I ask something?
Yes, yes.
Yes, sir. Sir, my name is David Vashne. So I'm asking something about that molecular docking and all.
Okay.
Yes, in previous time, I docked some with Autodoc Vina and used some in silico tools for that the drug discovery and development. So that tools are good for that drug discovery or not?
Well, see, whenever we are using any tool, it's a kind of a general recommendation. As I said, if we are good at cooking, anyone can tell if we are good cook or not, right? First time when we are making chapati, we of course not be able to make perfect round chapati. So if we are making food, it's like just making food actually. So we can evaluate if we have made a good food or not. Likewise, every tool which we are using, right? So we have to validate it, of course. And no tool is perfect. Every tool has its own limitation, advantages, disadvantages. Some of them they work well, some of them they do not work well. Even the commercial tools, they are not perfect actually. Even all those commercial tools, I don't want to name them, but those are not perfect. So they may work in some areas, they may not work in some areas. Likewise, open source tool, unless we know the theory behind the tool which we are using. If we understand how it is doing, then we are good to go, because then we can say that okay, this is reliable. Otherwise, just using anything blindly is not a good idea. It's not even an idea.
Okay, sir. I understand. Thank you, sir.
I think we can wind up this session because it's more than.
Sure. I just the last slides to thank everyone. Okay, last slide should.
So after this, an AlphaFold session will be coming soon. So stay tuned and please do follow us on Instagram, Twitter, and YouTube and LinkedIn also. Thank you.
Please come quickly. Snacks are getting cold.
Thank you everyone for joining this event. It's very pleasure to invite you here. So let's wait for you. Let's meet in an episode 8. Bye everyone. Kindly fill the feedback form. Bye. Thanks for joining.